the search of starch in food. In order to carry out this experiment, we need the following materials and tools: some glucosiodine, distilled water, starch, various food such as potatoes, sugar, milk, and biscuits, test tube and test tube dregs, glass roots and droppers, mortar and pestles. Objective: to determine whether the organic sample examined contains starch. Method: first of all, we have to prepare the two reference samples. The first one only with distilled water and glucosiodine, and the second one with 3 or 4 drops of the reagent, a small amount of soluble starch, and few milliliters of water to create a solution. The first one will have a light brown color, and the second one will have a blue color. After creating the two reference samples, we have to crumble finally each solid food in the mortar and to put them in the test tube marked with the corresponding name. Then we have to add from 5 to 10 milliliters of distilled water so that all the test tube reach the same level. Then we have to mix well and add from 3 to 4 drops of glucosiodine in each test tube. Finally, we have only to compare the result of each test with the reference samples. Then we have only to repeat the same procedure for all the foods analyzed. There we can see milk with glucosiodine. Sugar dissolved into distilled water and glucosiodine. Then biscuits are crumbled and dissolved into distilled water and glucosiodine. Conclusions. The two tubes containing biscuit and potatoes have got a blue color because there is starch in them. The test tube that contains milk has got a brown color because milk is made up of lactose, a carbohydrate composed of glucose and galactose and not of starch. Also, the test tube with sugar has the original coloration of glucosiodine because sucrose is made of glucose and fructose, so there isn't amylose in it.